So thank you very much for inviting me. I'm going to present a very different story than the much more technical talks you heard earlier. I'm actually going to tell you a story about how we've been wrestling with trying to understand the association between genotype and phenotype. And if I think back to how I got involved in biomedical research, this was really one of the key questions. How does our genome determine who we are? So one of the great things about working in biomedical research today, and you've heard this underlying all of the talks so far, is the idea that we're just awash in data. And I like to use quotes in my presentations that capture some of these ideas. So this is actually one of my favorites. This is a quote from Jules Poincaré. "Science is built with facts as a house is with stones, but a collection of facts is no more a science than a heap of stones is a house." And for me, this really captures the essential elements of what we struggle with in doing biomedical research, that we have access to far more data today than we could ever imagine. But the challenge is to take that data and to put it in a framework in which we can interpret it. And from my title, you'll notice that probably I'm going to talk a little bit about networks and how we use networks. And I'll just start out by pointing out that we have actually developed a lot of methods over the last few years for modeling networks and the associations they represent in a biochemical and cellular context. So we tend to name them after animals. So if you look along the right-hand side, you'll see all the animal names. We refer to our group of algorithms as the methodological zoo. You might point out that monster is a bit of a stretch as an animal. It's an animal. But all these different methods were developed to address slightly different questions and often to take advantage of different data sets. So people will often ask me, well, why don't you incorporate this into that model? And the answer is typically that we don't have data sets that have that and the other things we want to put together. So we have a collection of models. They're all open source tools. They all have GitHub homes. And whenever we publish any of these models or any of these methods, they first go up on archive or bioarchive, and the software is released on GitHub. So if you're interested in any of these, feel free to drop me a note if you have particular data sets or data types you want to look at. I'll point out that Panda has actually been named by the guy who's developing it, or renamed Spider. So if you ask me about Red Panda, I'll talk to you about Spider. But today I'm going to talk about this method we developed called Condor. And Condor was created to really address one of the key challenges that we were facing in trying to understand how genetic variants influence phenotype. And it's based on the idea of representing a network. So I know people in this auditorium have lots of different backgrounds, and so I was just going to start with a couple core ideas. And the first is that biological systems are driven by complex networks. And that's our underlying thesis, and I'll talk more about why we believe that's true later. But the structure of the network is fundamentally important. And the structure of that network informs our understanding of the system that we're trying to study. So people argue with me about this, but I would say that your network in your liver cells is different than my network in my liver cells. Although we could look at all the people in the room and come up with a canonical network that represents something about our liver cells. Healthy livers and diseased livers are going to have slightly different networks, and each individual is going to have a slightly different network. So when we talk about networks, we often fuzz over that fact when we talk about the aggregate networks. But the important thing, whether you think about aggregate or individual, is that networks differ between different states. And in the same way we think about differential expression, we translate that to thinking about differential regulation or differential network structure. So I would argue that there's no single right network. The network in each tissue in each state is distinct. And again, people have argued with me about that. This is going to be my operational definition of what a network is and how we use a network. And when I say I'm going to represent a network, I'm going to be honest with you and tell you that lots of people who draw networks and talk about systems biology draw different types of networks. A network is simply a way of representing complex association patterns between entities. And if you think about sort of a canonical network that people like to draw, they're balls and sticks. And mathematically, when we take a network like this, we represent it as an adjacency matrix. So what this shows, for example, is entity 1. It could be a protein that's interacting with itself and with other proteins, or it could be a gene that's regulating other things and regulating itself. But in this case, what it says is 1 interacts with itself, with 2, and with 5. And if I look up here, this row says 1 interacts with 1, with 2, 3, 4, and with 5. So There's a mathematical representation of any network as a matrix, and there are lots of subtleties as how we represent those. But the important thing to remember, too, is a network can actually represent a wide array of different things. And people often gloss over this, but it can be physical interactions between entities, like proteins binding to each other, genes interacting through a regulatory process, genes and metabolites working together, or it can be a representation of associations, statistical associations, say, correlations between the expression profiles of genes. And so we have to keep in mind what we mean and when we talk about networks. And the first networks I'm going to talk about, the only networks I'm going to talk about today in the limited time I have, is really a set of associations that we discover between different objects, between SNPs and genes. So some background. Why are we trying to do this? Well, when I think about genetics, I look back to the last biology course I ever took, which was in high school. And when I learned about genetics, I learned about Mendel, and I learned about peas, right? And if you think about Mendel's idea of genetics, it was really simple. We have genetic variants, and those genetic variants determine traits, which we all believe. And then those traits are assorted through breeding or through reproduction and are passed on to other generations. But the fundamental idea is that we have different variants of genes, and those different variants of genes represent the potential to develop different traits. So this is a really nice, simple model, right? Our genes determine our traits, and it's at the core of most of what we do. And when we think about looking at biological systems, that's most of what we think about. In the modern language of looking at the entire genome and all of the genetic variants, you can modify this to say, well, the genotype determines your phenotype, right? And again, that's something that we all believe in a very fundamental way. But the challenge is, as we've started to do modern genome analysis, really launched by the sequencing of the human genome in 2000, we've been able to catalog genetic variants. We've been able to identify millions of common variants that distinguish my genome from your genome. And we've had this idea that by mapping common variants, we could identify those variants which are associated with individual traits. And this is what I refer to as the GWAS puzzle, the Genome-Wide Association Study puzzle. The idea is actually very fundamentally simple, that what I'm going to do is I'm going to look for genetic variants at some position. So I'm going to look for a SNP. And here's some position in the genome where the SNP can either have a G or an A. And then I'm going to look at two populations. I'm going to look at a healthy population and a diseased population, or a tall population and a short population, or whatever you like in terms of different phenotypes. I'm going to look at those populations, and I'm going to try to find genetic variants that are more common in one than the other. So here you might be able to see there are more green genotypes than there are in the control population. And so the suggestion from genome-wide association studies would be that this variant might be important for determining whatever trait it is that we're looking at. Now, that's a really nice idea, and it's one that's led to some very important discoveries. But in fact, when we look at most complex traits, what we discover is that it's not one or two variants. It's not what Mendel would have thought of as being genotype determining phenotype. But instead, what we see is there are often thousands of variants that determine traits. So this is one of my favorite papers over the last few years. This is from a consortium called GIANT. You may not be able to see it, but they analyzed about 250,000 individuals, and they looked for genetic variants that were associated with height, right? You probably guessed from looking at me that I did not come from a tall family, for which I curse my parents. Genetics does influence height. That's something we know. So when this group looked for the genetic associations of variation with height, what they discovered is that there are about 700 genetic variants that explain 20% of your height. To get to 21%, they needed almost 2,000 variants. To get to 24%, they needed almost 4,000 variants. To get to 29%, they needed almost 10,000 variants. Okay? At some point, the genetic influence starts to drift away. It's hard to imagine how we account for all these small effect size variants. Is height alone? No. You can look at BMI. This group did a follow-on study, this time with 340,000 people. They said that 97 SNPs explain 3% of BMI, and that all common SNPs, so all 6 million SNPs, may explain 20% of BMI. All right? What do? Do we just give up on genome-wide association studies? Do we find map everything? Or do we think about this differently? Well, people have argued that we should think about fine mapping, that we should look for rare variants that somehow influence traits. So again, this is one of my favorite studies over the last few years. This was a really nice paper looking at type 2 diabetes. And the conclusion is that large-scale sequencing with 111,000 individuals, large-scale sequencing does not support the idea that lower frequency variants have a major role in disposition to type 2 diabetes. Okay? So the conclusion is basically that rare variants are dust. And there's a nice little figure in the paper that sort of shows different modeling approaches which all have a much greater estimate of the effect of these genetic variants than anything they see in their underlying data. Okay? So people have said, well, maybe it's just type 2 diabetes. Maybe rare variants are important in other diseases or other traits. I think we're trying to push the model too far. I think it's time to ask, can we think differently about this? So we've approached this problem by looking at genetic variants and asking ourselves, can we take genetic variants, which have been found through genome-wide association studies, and provide links to something which gets us close to the phenotype? And the closest a few years ago I could imagine getting to the phenotype is gene expression. It's something we measure pretty commonly. So we decided to do an eQTL analysis, an expression quantitative trait locus analysis. Now, if you're not familiar with QTLs, QTLs was a technique that was developed in the 1970s by Steve Tanksley, who was working on the medically important problem of, anybody? Tomato breeding, right? So he was looking at quantitative traits like the redness, the roundness, the ripeness of fruit, and trying to associate genetic variants he could measure with those quantitative traits. Now, about 17 or 18 years ago, a couple groups independently recognized we could take all of the genes in the genome and treat those as 25,000 or however many genes you believe there are quantitative traits. So we can do an expression quantitative trait locus analysis, and we look at associations between genome-wide genetic variants and the expression of individual genes. So we treat gene expression as a quantitative trait, and we simply ask which SNPs are correlated with the degree of gene expression. And when most people do this, they look at SNPs which are immediately adjacent to the genes, what are referred to as cis-acting SNPs. What we decided to do was to take into account both SNPs which are immediately adjacent to genes as well as those that are far away, the trans-acting SNPs. And there's a growing body of evidence that suggests that many of these faraway SNPs are actually important for phenotype. So we decided to put all this together and just do a really simple analysis. So there's a great tool called Matrix eQTL that we used. And what it does is it looks at each gene, and then it looks at each location where we measure a SNP genotype. And it looks for a correlation between the expression of gene 1 and SNP location A. And it does that for every gene. It does that for every SNP. So 25,000 genes, 6 million common SNPs on most of these genotype chips. And so we get a whole set of associations. And what we do is we look for those associations which are statistically significant, and we collect them. And the big trick behind Condor, this method we developed to look at eQTLs, is that we decide to treat these, to represent these, as elements of a bipartite graph. So we represent these as a network. What's a bipartite graph? The easy way to think about it is that it's a graph that has two types of nodes. So I've shown them as squares and circles, or SNPs and genes. And the only rule about a bipartite graph is that elements of the same type are not linked to each other. So the only links here are between SNPs and genes. And what it does is it represents the many-to-many associations that come out of an eQTL analysis. So I have one gene that seems to be influenced by many SNPs, and one SNP that seems to influence many genes. So what this allows us to do is to capture those associations. And then what we asked ourselves is whether or not the structure of this network actually told us anything meaningful about the underlying biology of the systems that we were trying to study. So we published this in PLOS Computational Biology now about 2 years ago. And it was a very interesting study looking at chronic obstructive pulmonary disease, COPD. So we had a data set in which we had a number of individuals who'd been genotyped. We also had gene expression data. And we did this kind of analysis and we drew a graph. Now the truth is we never draw graphs. If you give a systems biology talk, you have to show a hairball. I was too lazy to actually draw one because they're uninformative. This is just some random one I grabbed off the internet. I don't know if it's genetic data or internet connectivity or the Russian troll data from Facebook that shows all their interactions in the last election. I have no idea what it is because most of the time these things don't tell us anything. But if you look at the properties of the graph, you actually learn something interesting, right? So we have a bipartite graph, and what I'm showing is the degree distribution for the SNPs and for the genes. And there are many more SNPs than genes, so I represent those separately. We talk about the out degree for the SNP and the in degree for the genes. And the reason we separate them is that there are many more SNPs than genes, but both of these represent what we call the degree distribution. So this is the log of the frequency, and this is the log of the degree. The degree is just the number of connections that an individual element has, and the frequency is the frequency. And the good thing is we see this kind of straight line behavior in both the SNP out degree and the gene in degree. And this is the kind of degree distribution we expect from naturally evolving networks, right? So we see the same kind of distribution for the internet. Up here is your laptop, and there are lots of those. Down here are Google and Amazon and eBay and Facebook, right? The things that are connected to everything. So these are the leaves in the network. These are the hubs. And what we know is there are many, many more leaves than there are hubs, right? And the hubs are fundamentally important because if you close your laptop, because I'm so interesting, you're actually going to close the laptop and listen. If you close your laptop, nobody but maybe your mother knows because she's trying to contact you on Skype. But if Google or Facebook shuts down, everybody freaks out, okay? Because they're holding the network together. So what we have are degree distributions that make sense in the context of what we'd expect. But then you can ask the question, are these informative? So I'm going to focus on the Snip degree. And what I'm going to do is I'm going to take GWAS-associated SNPs that have been linked, and I can do this for COPD, I can do this for all pulmonary diseases, I can do it for all diseases in the NIH GWAS catalog. The story is essentially the same. Down in the bottom, we have the hubs, and we're going to project GWAS hits onto this graph. And the question I have is, where do you expect the GWAS hits to land? And the answer is, when we project them, you'll see little red spots appear. And I don't know if you can see them or not, but if you watch the top left-hand corner, you'll see that they all appear there. The hubs are a GWAS desert. And that really puzzled us initially, that we don't see genetic variants associated with disease falling in the hubs. We actually see a little bit of an overrepresentation in the middle and underrepresentation at the top. But the GWAS variants fall in to avoid the hubs. Why? Well, we think this is actually survival bias. That if you land in the hubs, you're too disruptive, and so you don't get seen. And a great example of that is what happened in World War II. The British were sending bombers to drop bombs on Germany. The planes were coming back shot up with holes. The RAF commissioned a study to map where those holes were with the thought that what they would do is build armor and armor the planes. And then Abraham Wald walked in one day and looked at what they were doing and said, that's stupid. Put the armor where the bullet holes aren't. Because if you shoot the engines or kill the pilot or knock out the fuel tanks, you don't see the bullet holes because the planes don't make it back. Part of the reason we think we don't see hubs with genetic variants is that they simply don't survive. So we asked ourselves, can we put this idea to work? Can we start to think about what this tells us about the structure of the network? And an interesting observation is that we had an overrepresentation in the middle of the graph. And what that suggested is that the network has a community structure. All networks tend to have community structure, right? You have a mobile phone, you can call anybody in the world. I can call my wife calls my son calls his grandmother, my mother calls me. And while I may one day call you, right, my wife or son or grandmother, my mother are never going to call you, right? So I have a family community that's defined by an increased likelihood that any two members of my family will call each other on any given day relative to the likelihood they're going to call you. Okay? In the same way, I have a social community, I have a work community. So there are methods in network physics for identifying communities. They're called generally modularity maximization. And what you find is a set of things that are more connected to each other than you'd expect by chance. So when we run this now on the EQTL network, what we find is a very highly modular structure. Right? So now this is representing the network as an adjacency matrix. And what you can see is they're very dense, very tight modules. There are some hubs, and you can see those in places where you see little vertical lines. But most of the SNPs and genes are organized into these tight communities. This is a different representation of those same networks. I think this is the Cshell representation. And what's interesting is when we look at those communities, particularly in the giant connected component, the biggest set of stuff that's connected all together, what we find is there are 34 communities, many of which are enriched for specific Go terms. And this is the place we start to get very interested, because what we have is not a single SNP regulating a single gene, but a family of genetic variants that regulate a process. So we got very excited because we're starting to move toward that idea that many SNPs can work collectively to try to influence different properties. In looking at these, though, what you also see is there's some internal structure in each of these, and there tend to be highly connected SNPs. And we call those core SNPs. And what we actually did was we developed a metric we call the core score for identifying those. And it's basically the fraction of modularity that's covered by an individual SNP. So we asked, do these core scores actually reflect known biology? So we took a comprehensive study, meta-analysis that identified 34 COPD-associated GWAS SNPs, and we asked, where do they land? The first thing we noticed is they land in communities that have relevant biological functions. But the second thing we noticed that was really exciting was when we looked at them, the average core score for those SNPs was 20 times higher than the average core score in the network. So not only is a family of SNPs associated with perturbing a process, but the likelihood that you're found in a GWAS study of being associated with disease has to do with how central you are to that process or how strong your relative perturbation might be. Really interesting finding. Is this generalizable? Well, we actually published a paper at the end of last year looking at 13 different tissues, and I'm going to get the hook in a minute if I don't move. But the short answer is yes, we see exactly the same thing in other tissues. So these are just a degree distribution I showed you earlier with the SNPs associated with disease up on top. What we see is there's a significantly higher core score for disease-associated SNPs within tissue, across tissue. And I'll just point out that we see that across all 13 tissues that we could analyze using data from GTEx. So what we've discovered is something very interesting. In most instances, it's not a single gene controlling a single trait, but a family of variants that perturb a process. Disease-associated SNPs map to communities whose genes have functions that make biological sense. The core SNPs are far more likely to be disease SNPs. And again, that makes sense. A global hub that perturbs things badly might kill you. On the other hand, the reason you develop a chronic disease probably has to do with the fact that there's a process in your cells that's not functioning appropriately. And the structure of the network is informative in ways that individual SNP gene associations are not. And the last piece, which is in that PNAS paper, actually has to do with tissue-specific processes and tissue-specific communities. And the really cool thing in that paper is we discovered that those tissue-specific properties nucleate around SNPs that sit in tissue-specific open chromatin. So I've run out of time. I've tried to tell you a really complex story in a short time. And so I want to close with a quote from Enrico Fermi, physicist like me. Fermi said, before I came here, I was confused about this subject. After listening to your lecture, I'm still confused, but at a higher level. So I hope I haven't left you highly confused, but I want to thank you for your patience. I know I'm standing in the way of lunch. Absolutely. $25 a download.
So I got a question.
Please.
So you suggested that like you would have studied the German down rankings, the English instead of studying actually know where to put the armor. Well, why don't we do the same thing development.
I mean, we generally don't see those, right? You have lots of spontaneous abortions.
No, but maybe you can.
There are lots of things you could do, right? It's just having access to the right data and the right samples. Please.
Next, so I was wondering, what is the difference between...
So what's the difference between core and not? So the core SNPs, and I sort of rushed through this, but we define the core, they have a high core score. And the core score is defined as the fraction of the modularity that's carried by an individual SNP, right? So they're like local hubs. And the way I always describe them is, if you think about my example of a family calling each other, right? If you have a big extended family, there's probably a matriarch in the family that holds the family together, and two warring fractions that are only held together because, you know, grandma is still alive, and if grandma dies, the family breaks into warring factions, right? It's the same kind of thing. These core SNPs are the ones that really hold the communities together, and if you disrupt them, you can really perturb the function of that community, right? I think this is really just a different way of looking at these associations, and while I can't tell you the networks are right, I can tell you they really give us a lot of insight into how these interactions might influence the phenotype.
So some SNPs are synonymous, like if you, and some are not, and like distinguish those.
So you can build an analysis of synonymous and non-synonymous SNPs, but most of these SNPs are actually non-coding SNPs. So we think they play some regulatory role, but most of these are non-coding. The number of coding SNPs are very, very small.
How about the transcription factor binding site?
Great question. So if you look in the very first paper, what we show is the core SNPs, actually based on regulome DB, have very, very different functional associations than the non-core SNPs, right? Which is an interesting observation. When we looked at the 13 different tissues in the PNAS paper, we found that, again, there are differences between the core and non-core SNPs. But there are core SNPs that are tissue-specific, and those appear in tissue-specific open chromatin. So they're really nice pieces of this picture that start to emerge from looking at these networks, right?
Anybody else? All right.
All right, thank you very much.